Good afternoon, and welcome to my presentation video. My name is Supatpong Juntarawatt, and this is my project. It is widely known that water is vital for living things and the environment. Yet many water sources are polluted from human actions. One of the causes is the oil spill from factories, increasing the overall temperature and destroying biodiversity in the water. Another potential damage comes from the dye. Even though it may seem not so dangerous, the dye is carcinogenic and hazardous to the environment. To relieve this problem, we propose the organic adsorbent from carbon aerogel, which has high porosity and hydrophobicity from cattle flowers. In order to create the aerogel, we start with putting the flower into a hydrothermal process at 120 degrees Celsius for 2 hours. After that, the sample is freeze-dried for another 24 hours before polarizing it at 800 degrees Celsius. Alternatively, we dope the final product with hydrothermal for 14 hours again, but this time, titanium dioxide pre-secure solution is used instead. For the characterization part, we use XRD, SEM, FTIR, and BET to analyze the structure, external morphology, existing chemical bonds, including the pore size and surface area of the final product respectively. From the resulting graph, we can see that the results show that both samples present peaks at around 28 and 41 degrees. Additionally, the peaks on carbon aerogels are broader than the doped one. This can be implied that the structure of our undoped aerogel is more amorphous than the counterpart. Since our microscope also has a function of EDS, which can tell the composition of any elements in the sample, we can report that our adsorbent consists of carbon, oxygen, silicon, and titanium. However, the exact proportion of each element is not confirmed, due to the fact that we use carbon tape for gluing the sample to the tool, making the figures vastly overestimated. These are the pictures from SEM under 5,000 and 10,000 times magnification. All of these show that the structure of doped aerogel is rougher than the other one. With more magnification, we can predict that titanium dioxide is attached on the surface, creating a cauliflower-like surface. We found also from FTIR that both of our carbon aerogels are composed of both methyl, ether, and hydroxyl functional group. Double bonds of two carbon atoms are also identified from the analysis. This is because that there are visible slight drops at the distinct wave number of each type of bonding. From the BET analysis, we found that our doped product consists of both macropores and mesopore, while the undoped ones have only macropores. After doping with titanium dioxide, our material has a higher surface area, lower pore size, and density. This behavior supports the higher efficiency adsorption in our aerogel. For our absorption efficiency, we demonstrate by pouring 20 ml of toluene and 0.5 grams of fresh cattle flowers or carbon aerogel into a closed petri dish. After 2 hours of adsorption, the samples are wet for 2, 5, and 10 minutes before weighing the final mass. We quantitatively analyze the result by comparing the missing mass during the adsorption to the initial mass. Due to their high hydrophobicity, the aerogel and the flower should be great oil adsorbents. We found that the aerogel is 3 times better than fresh flower for oil adsorption. In order to find the efficiency in methylene blue adsorption, 0.1 grams of sample was mixed with 100 ppm of methylene blue under 200 rpm stirring rate. After 75 and 150 minutes has passed, 1 ml of the solution was diluted and centrifuged to determine the final concentration. In fact, the concentration of the dye by the adsorbent of this solution at a wavelength around 660 nanometer by UV with spectrometer. As a pre-analysis result, we can conclude that the relationship between the adsorbent A and its concentration C is A equal 0.141 C minus 0.1099 from the calibration curve. Similarly, the methylene blue adsorption shows that the aerogel has 3.5% higher efficiency than the original product. In the end, we should say that our final product can absorb oil and dye better than the original study material, which are the flowers. Thank you for your attention.